Okay, students, so this is Industrial Biochemistry and the course code is BCHM-351. This is lecture 4 and the topic is continuous and batch type culture techniques. I am Dr. Sadiq Ibrahim, Assistant Professor, Department of Chemistry, Guwahati University of Science and Technology, and you can access all these lectures by using the Trust Media Server portal, which is www.ms.guwahati.edu.pk. Now, the topics which we are going to discuss in this lecture, they include the fermentation techniques for growing cultures. First one is the batch culture. The second one is the continuous culture, and the third one is the fed batch culture. Now, the fermentation techniques for growing cultures. So, the cultures can be grown in three classical operational modes, which are batch mode, continuous mode, and fed batch mode. Now, for the past 3 decades, there has been a tremendous growth in the use of fed batch reactors in fermentation technology. So it means that the fed batch mode is preferred over continuous mode or batch mode in the industries. The biotechnology, chemical and pharmaceutical industries, they prefer to use the fed batch process due to certain advantages, like it is economical, the yield is very high in case of using this fed batch mode, and it is also very good to be used. Now, the fermentation techniques which are used for growing cultures, these are the batch mode and fed batch processes, and these techniques are used to handle normally low volume and high value products of the fermentation process. So these include amino acids, antibiotics and chemical compounds which are produced as a result of using either batch mode or fed batch mode in the fermentation technology, and as a result of this the products which are obtained normally, which are either primary metabolites or secondary metabolites of the microbial cells, so they are of low volume, so the end product and the yield is low volume, but they are high value products. As you can see that the antibiotics nowadays are very expensive. Similarly, the amino acid supplements are also very expensive. Now, due to the high values of these products, the profitability can be improved greatly by optimization and improvements in the yield and productivity. So it means that during the process of fermentation, if the industrialist is using either batch or fed batch process, so by optimizing the conditions, either if you are talking about the nutrients addition, or you are talking about the pH optimization, or you are talking about the air aerobic process optimization, so by optimizing all these processes, the yield as well as the productivity of these high value products can be increased. Now, first of all, we will be discussing the batch culture. So in case of batch culture within a fermentation process, the all necessary medium components, the nutrients, along with the inoculum, which is the starter culture, are added at the beginning, which means at the start of the fermentation process, and not during period of fermentation. So this is important to note that in case of batch culture, the medium and the inoculum both are added at the start of the fermentation process, and no addition of medium or inoculum is taking place during the process of fermentation. Now the medium concentrations are not controlled but are allowed to vary as the living cells take them up. So it means that the nutrients in the form of yeast or lactose, which are provided as a food for the microbial cells, so they are not being controlled, which means that they are not being removed either or being added, but they are allowed to change as the microbial cells are utilizing them for their growth and reproduction purposes. Now the products which are normally the primary metabolites or the secondary metabolites, whether they are intracellular or extracellular, are harvested only at the end of the run. So this is important to note that when the fermentation process has completed, then the high value products are being collected or harvested, and then they are being refined and purified. So in batch culture, the basic controls for pH, temperature, and dissolved oxygen are applied during the course of the batch culture within the fermentation tank during the process of fermentation. So it means that uh, the optimum levels of the pH, temperature, and the oxygen are maintained during the process of fermentation in a batch mode. Now, the pH, the dissolved oxygen, and the temperature, they are normally held constant during the course of the batch reactor operation. Now, the only optimization parameters are the initial medium composition. So the optimization is carried out at the start of the batch culture within the fermentation process, and that is the medium composition, that how much lactose or yeast or corn steep liquid needs to be added to the culture media. So that is the only optimization parameter, and which is set at the start of the fermentation process. Now moving towards the continuous culture. So in case of continuous culture, one or more feed streams containing the necessary nutrients or media are fed continuously, while the effluent stream containing the cells, products, and the residuals is continuously removed. So by noticing this, you can understand the basic difference between batch culture and the continuous culture. So in case of batch culture, the media is introduced into the fermentation tank at the start of the fermentation process, and the high value products or the cells are collected at the end of the run. While in case of continuous culture, the nutrients are being added continuously during the whole run of the fermentation process, as well as the outlet present within the fermentation tank uh, takes up the effluent stream, which is containing the cells, the high value products like primary or secondary metabolites, as well as the residuals. So they are continuously removed along the whole run. Now, as a result of this, a steady state is established by maintaining an equal volumetric flow rate for the feed and effluent streams. So it means that as much the uh, nutrients are being added, so much are the effluents being removed from the fermentation tank during the process of fermentation and during the whole run. Now the culture volume is kept constant and all the nutrient concentrations remain at constant steady state values. So it means that if you are taking out, let's say, 1 liter of the harvested material from the continuous culture batch fermentation tank, so you are adding the same amount of nutrients to the fermentation tank as well, and this process is going on till the very end of the fermentation run. Now, in case of continuous cultures, the continuous batch operations are more common in the chemical industries. Now, with the exception of single cell protein production, certain beer production, and municipal waste treatment processes, the continuous culture batch mode has not been adopted widely by the industry. So it means that most of the industries which are adopting the continuous culture mode technique are the chemical industries, the single cell protein production industries, the beer production industries, and the waste treatment plants of the municipalities. Other than that, all the other industries like pharmaceutical industry or the cosmetic industry, they are adopting the batch mode or fed batch mode. Now the continuous culture, it is not a dominant mode of industrial operation, primarily because of the difficulty in maintaining sterility, free of contamination by other organisms, as well as the protection against phage attacks. So there is a danger of contamination in case of using continuous culture mode, as well as there is a danger of phage attacks, that is the viral attack within the continuous culture mode, because during the whole process, during the whole run of the fermentation process, you are adding nutrients as well as you are removing the cells and the products and the residuals. So during the process of fermentation, uh, the addition and removal, there is a danger that the contamination of other microorganisms can take place within the fermentation tank, as well as the viral attack can also take place. So as a matter of fact, the steady state operations are found to yield poorer results than dynamic operations like batch mode and fed batch mode. So there is an edge or advantage of batch mode and fed batch mode uh, on top of the continuous culture, that the yield, the product yield, the percentage of the high value product obtained is higher in case of batch mode and fed batch mode in comparison to the continuous culture technique. Now moving towards the third mode, which is the fed batch culture mode. So in case of a fed batch culture mode, it is a type of a semi-batch operation in which the nutrients which are necessary for the cell growth and product formation, and the product here means the primary and secondary metabolites. So the nutrients are fed either intermittently, that is after a certain period of time, or continuously via one or more feed streams during the course of the operation. So it means that during the whole process of fermentation, from the start to the very end, the nutrients are being uh, continuously added or intermittently added to the fermentation tank. Now, the culture broth is harvested or collected usually at the end of the operational period. So when the fermentation run has completed, then the collection of the high value products and cells is carried out. Now, there is a option that either fully or partially. So whether the harvesting is carried out fully or partially. If it is carried out partially, then the remainder serving as the inoculum for the next repeated run. So the rest of the media or the culture which is left within the fermentation tank, it is used as a starter culture for the next repeated run for the fed batch culture, and that is why it is called fed batch culture, that the feeding is being carried out continuously throughout the fermentation process. Now, in case of the fed batch culture, the process may be repeated, which is also known as repeated fed batch, a number of times if the cells are fully viable or active and productive as well. So it means that at the end of the first run of the fed batch culture, some of the harvesting is carried out. So some of the material or the media from the fermentation tank is carried out, and the rest is left within the fermentation tank, and that is a repetition, but provided that the microbial cells are active as well as productive, that they can produce the primary or secondary metabolites. So there are one or more feed streams but no effluent during the course of operation. So it means that you are adding the nutrients to the fed batch culture during the whole process of the fermentation technique, but you are not taking out any effluent during the fed batch culture, but you are only taking out the effluent stream at the very end of the fermentation process. Now, there is also the possibility that by manipulating the feed rates, that is either decreasing or increasing the amount of nutrients to be added to, to the fermentation tank, the concentration of the limiting nutrients in the culture can be manipulated either to remain at a constant level or to follow a predetermined optimal profile. So depending upon the nature of the fermentation technique and the nature of the high value product which is targeted to be obtained, the nutrients amounts are either increased or decreased within the fermentation tank, and as a result the yield is manipulated. And if it is not working or it is not favored, then a predetermined optimal profile can also be followed, which is written in the manual. So in case of the fed batch culture, as a result the culture volume reaches the maximum as a result of repeated addition of the nutrients during the whole course of fermentation. Finally, a batch mode of operation is used to attain the final yield. So a batch mode of operation means that at the end the harvesting is carried out and the end product is being collected or gathered, so for further purification of the primary or secondary metabolites as the case may be. So by looking at the fed batch culture, the continuous culture and the batch culture mode, you can see that what is the main difference between these three modes during the fermentation process. So in case of the batch culture mode, you are adding everything at the start of the fermentation process, and then everything runs smoothly, and after the specific period of time when the run has completed, so then the harvesting is carried out. Now in case of the continuous culture mode, there is a continuous addition of nutrients through the whole run of the fermentation process, as well as there is a continuous removal of the effluents from the fermentation tank during the whole process of the fermentation technique. Now in case of the fed batch culture, it is more like a batch culture mode, but the only difference between the batch culture and the fed batch culture is that the fed batch culture is the feeding of the nutrients either continuously or after a small period of time intermittently to the fermentation tank. So you need to remember that in case of the fed batch culture, the nutrients are being added through the whole run of the fermentation technique, but there is no removal of the effluents from the fermentation tank. Now the references which need to be followed for further understanding and for further reading are mentioned here, and the serial number 5, the book with the title Principles of Fermentation Technology, is very relevant to this topic, and it is written by Peter F. Stanbury, Alan Whitaker, and Stephen Hall. So if you want to further grasp the idea of continuous and batch type culture techniques in fermentation, so this book is very relevant for understanding and clearing your concept. Thank you.